Hello, everyone. My name is Mark, and welcome to this new episode of the mitoPodCast. For those of you joining us for the first time, the mitoPodcast is a new program that we hope will raise awareness in the mitochondrial medicine, research, and patient advocacy community on topics related to mitochondrial disease and health, as well as to highlight cutting-edge research in the field. Our audio format episodes are approximately 10 minutes in length and will feature the exciting work of an expert in the field of mitochondrial medicine in a way that can be understood by all in the community, regardless of educational background. Included with every episode will be an infographic summary of what was discussed, so please check that out as well. For this episode, I am delighted to be joined by Dr. Neal Sondheimer. Dr. Sondheimer is a staff physician in the Clinical and Metabolic Genetics Division at the Hospital for Sick Children in Toronto. He is also an associate scientist with the Genetics and Genome Biology Research Program at the SickKids Research Institute. Furthermore, Dr. Sondheimer is an associate professor with the Department of Pediatrics at the University of Toronto. He completed an MD PhD at the University of Chicago, followed by clinical training in clinical biochemistry, genetics, and pediatrics at the Children's Hospital of Philadelphia. Dr. Sondheimer, it is my pleasure to welcome you to the mitoPodcast.
Thank you, Mark.
Dr. Sondheimer, your research program has investigated mutations in the mitochondrial genome that have been closely associated with a wide variety of disorders. These mutations are said to exist in a state of heteroplasmy. Could you explain to listeners what heteroplasmy is? And how do we know if a mutation is pathogenic, meaning dangerous for cells?
So heteroplasmy is actually a confusing concept even sometimes for geneticists. When we think about how genes work, we generally think of genes being present in 2 copies each in the cell, and most people only have disease when both copies are mutated. But that's not how mitochondrial DNA works. There are several differences that are important. One critical difference is that in general all mitochondrial DNA comes from your mother. But the other key difference is that the number of genomes in every cell can be different. So there's not exactly 2 copies of DNA. There can be hundreds or even thousands of copies present. And as a result, mutations build up as a ratio rather than fixed intervals. So heteroplasmy refers to the presence of 2 different allele copies of mitochondrial DNA within a cell or within a person. And heteroplasmy is important in disease states because the more pathogenic mitochondrial DNA you have, generally the worse your condition is, whereas the less you have, the milder the symptoms, and below a certain threshold, no symptoms exist at all. In terms of understanding how mitochondrial mutations can cause disease, this is a very important problem that we study both in clinics and in the lab. What we want to do is find out when a patient has a new variant that we haven't seen before, whether it's actually causing disease or not. We can do this in a number of different ways. We can actually do it by comparing the sequence with other people or even other organisms. And we can also do it by techniques in the lab, like actually kidnapping the mitochondria from patient cells and putting them into a different cell type to see if the mitochondria functions differently with a different sequence.
How can you alter heteroplasmy if it causes disease?
So the alteration of heteroplasmy is something that people have been thinking about for a long time. It's probably been about 25 years since the first papers were written about the idea of changing heteroplasmy. The idea is critical because if normal mitochondrial DNA is present even in our patient cells, if we can just make that mitochondrial genome take over the cell, we could possibly reduce the symptoms of disease or even eliminate it entirely. So in order to do this, we either need to make the bad copy of mitochondrial DNA replicate less or make the good copies of mitochondrial DNA replicate more. Most approaches to doing this have looked at actually cutting the mitochondrial DNA. So if you cut it in a sequence-specific fashion and get rid of only the bad copies, then presumably the good copies will take over the cell. And these approaches have been looked at in animal models and in cells and seem to work well, but haven't been used in patients yet. In our lab, we're looking at a different approach. For some mitochondrial mutations, there are actually physical changes in the mitochondrial DNA that occur that can be targeted with small molecules. And we're trying to use a class of small molecules to inhibit the replication of bad mitochondrial DNA and encourage good mitochondrial DNA to overpopulate. Hopefully, by doing this, we'll be able to improve the symptoms of disease in some patients with specific mutations.
Great. Thank you, Dr. Sondheimer. So what are some next steps you would like to take in this research, and how do you see this translating in diagnosis and treatment?
So some of the next steps we'd like to take, the project that I was just describing, we actually did it in patient cell lines. And the next step that we'd like to take is to actually see if we can do it in patients. This is challenging because doing clinical trials in patients with mitochondrial disease is always complex. It's hard to find sufficient numbers of patients. It's hard to find patients whose disease is exactly the same. It's also hard to structure clinical trials where you're treating some patients and not treating others, because sometimes that feels unfair. But these are the only ways we can really move forward in research, is by doing clinical trials. Hopefully, if we're able to find strategies to alter heteroplasmy, and probably we'll take more strategies than the one that I'm describing, eventually we'll come up with new classes of therapies that can be used for patients with mitochondrial diseases due to mitochondrial DNA mutations.
As you know, the development of new treatments for mitochondrial dysfunction is not easy. In your view, how could we move forward faster in a careful and responsible way that prioritizes patient safety? And are there any novel therapies that you believe show significant promise?
So in general, what we like to do with therapies is to try them in animal models first, to try to understand if they're going to be safe and effective. But as we're entering a new era of molecular medicine, we're reaching the limits of the ability to do that. Some therapies are so specific to patients and their particular mutations that there's simply no way to try them in animal models. And we're simply going to have to try them in patients first. That's always challenging because we worry about patient safety, and there have been negative experiences in gene therapy development where things have gone wrong in ways that couldn't have been predicted, and sometimes many years down the road. We partner with the health authorities, with Health Canada in Canada and the Food and Drug Administration in the United States, to design therapies that maximize safety. But it's also become apparent that we need to accelerate these therapies towards the clinic, because it's not fair to come up with concepts that may be useful in treating patients and not explore them. So there has to be a balance between the trade-offs of safety and the availability of new therapies, because really families demand it. We can't only focus on safety, because these diseases, many of them are fatal, and patients cannot wait until new therapies are introduced. These are difficult issues, and in the design of any trial, they come up all the time, and we try to do our best to balance known risk factors to patients with the potential for them to get advantages from new forms of therapy. In terms of novel therapies that may show promise, there are a lot of exciting opportunities. In general, gene therapies seem much closer now than they have in the past. And gene therapies will work for patients with mitochondrial disorders as they will for many other diseases. Although I work specifically on diseases due to mitochondrial DNA mutations, many diseases are due to mutations in nuclear genes that work inside the mitochondria. And for those diseases, the approaches are effectively similar. They can be more challenging because mitochondrial disease doesn't just affect one tissue or organ system, it affects really the entire body. And so we need to develop therapies that will target the brain and the heart, the kidney and the liver, and all the organ systems that are affected in patients with disease. But we're learning to do that. There's also been a lot of development at bridges between direct gene therapy, where we're trying to insert new copies of genes or repair genes that are broken. And these therapies could include the use of mRNA, which is basically a bridge. You give cells the information that's required to make proteins, but you do it in a non-permanent fashion. And this may have trade-offs in terms of safety and efficacy that may be beneficial for some patients. So I think those kind of therapies show a lot of promise. In terms of the mitochondrial function itself, a lot of the approaches that are being considered right now have to do with looking at mitochondrial quality control, so ensuring that the best mitochondria are present. And so the way those therapies work is that our bodies have a natural process of clearing mitochondria that are dysfunctional called mitophagy. And by increasing mitophagy, the hope is that we'll actually select for a better performing population of mitochondria, which will make cells healthier. The converse approach is actually simply ramping up the number of mitochondria that are produced. And so those therapies address something called mitochondria genesis, which is just the production of new mitochondria. And the idea there, and it's a bit of a reverse and almost a complement to the mitophagy therapies, is that if you have impaired function, it might be better to have more mitochondria present. So those therapies will ramp up the number of mitochondria and the mitochondrial mass that's present in cells, hopefully to improve patient symptoms.
Great. Thank you for that, Dr. Sondheimer. From the perspective of a research scientist and a physician, how do you think a network of patients, researchers, and clinicians collaborating together benefits mitochondrial medicine and research?
So the collaboration between patients and doctors and researchers is critical in all forms of rare disease, and is probably especially critical for mitochondrial medicine. Mitochondrial disease is challenging to think about because it's not one single disorder. So cystic fibrosis is a single disorder. It's caused by the same gene being affected in many patients. But in mitochondrial disease, you have many patients with different changes in different genes all coming together. That makes the disorders much harder to study because there are much smaller populations. It also impacts the voice of patients and the voice of clinicians and researchers by making it smaller. In order to work together towards solutions, we actually have to be well organized and work as a team to advocate for resources and to organize clinical trials and to know who each other are. One of the big advantages of working within a network is that we'll be able to find the patients when we need them. Patients will be able to find the clinicians when they're ill, and they'll be able to direct resources towards the researchers who are trying to develop new therapies for their children and family members.
Great. Thank you so much, Dr. Sondheimer, for coming on the program today and for talking about the extraordinary and cutting-edge research that you do. Before we wrap up this installment of the mitoPodcast, do you have a final message for the listeners? And where can they contact you if they would like to learn more about your work?
So I guess my final message is a continuation of the past answer, which is that you need to be involved. It's important to recognize that if you have a family member with mitochondrial disease, you really are part of a larger community, and that by working together with that community, you can achieve better results than you could working by yourself. And in terms of how to contact us, our laboratory has a website through the University of Toronto, and you can look up Sondheimer Lab, and the information is there and available.
Thank you so much, Dr. Sondheimer. Before we end for today, I would like to thank all our listeners for today's episode. If you have any comments or suggestions for future episodes, we would love to hear from you. Please head on over to our website to download our episodes and to see the upcoming events we have for this year. We have some exciting initiatives planned, so please stay tuned. Thank you all once again, and tune in to our next episode featuring another expert doing exciting work in the field of mitochondrial medicine.